So in mitosis, what's going to happen in mitosis is we're going to take typically diploid cells. We're going to copy everything in the diploid cell, chromosomes as well as mitochondria, chloroplasts if it has them, ribosomes, and we're going to take that and divide that into two cells. So you're going to go from one cell to two cells, and the two cells are going to be basically duplicates of each other. This occurs in basically most of the cells in your body. So in your nerve cells, cells in your intestines, skin cells, basically anywhere in the body, this is happening, this process of mitosis and making diploid cells. Just to make sure you understand, haploid cells, one set of chromosomes, those typically only take place, that process, we'll talk about that next, making haploid cells occurs in the testes and the ovaries in a human, and those result in sperm and eggs. So what we're going to talk about now, though, is this process of mitosis. And we'll start with talking about the cell cycle that we start with is interphase. And so an interphase, which represents about 90% of the time that the cell is in this cycle, includes G1, S, and G2 phase. The G can stand for gap or growth depending on the textbook you use. You see sort of both of those. And basically what happens in G1 phase is this is where things like the mitochondria and the ribosomes would be replicated. So that occurs during G1. That replication of mitochondria, ribosomes, endoplasmic reticulum, basically all the organelles except the DNA, that occurs in G1 through S phase and into G2. What's unique is in S phase, that's where the DNA synthesis occurs. And so during S phase, DNA replication occurs, and what happens there, that's where you take your DNA polymerase and you're traveling along copying all the nucleotide sequences in the chromosomes. So that's when DNA replication occurs during S phase only. So that represents interphase collectively, and again, that's 90% of the time that the cell's in that cycle. So then what we look at next here is, this is a cell, this shows you the cell in interphase, and once again, if you remember back, I told you the first time you see a cell, when you look at a cell, what you would see is you would not see chromosomes. Instead, what you would see are this strandy type of stuff, all these strands in here, and that's chromatin. And remember that chromatin is the DNA that's wrapped around these specialized proteins. The proteins are called histones, and so the proteins the DNA is wrapped around these proteins, and then that's wrapped up into this sort of string-like material that makes up chromatin. Also, what you'll notice in this picture is this thing called the centrosomes, which include these centrioles. So these are the centrioles here, these centriole pairs. Those are going to be important later on, but you'll see there's a pair of them, and those also get replicated. This is a typical animal cell that they're showing you during division here. So once again, right here, you see here's the nucleus of the cell, here's the chromatin, and then here's these centrioles here during interphase. During prophase, which is really the beginning of mitosis, what happens is the chromatin starts to condense, and it condenses into these chromosomes. So remember, at this point, the S phase has occurred, so the DNA is actually doubled. It has already replicated. So you end up with these chromosomes here, and in this particular cell, the way they've drawn these chromosomes, you'll notice that there's two big ones and two small ones, okay? So the N value of this cell is going to be 2, because that means there's 2 in a set. There's this one, and then there's this one. That's one set. But you know, it's a diploid cell, so it has 2 sets. So it's got one of those and one of those, but then it also has another big one and another small one. If this was a human cell going through mitosis, you would expect to find an N value of 23, and there would be 46 of these duplicated chromosomes at this point. Okay, so also what happens there is the nucleus starts to disappear, and you'll start seeing the formation of these protein fibers that we call the meiotic spindle. And you'll notice that those stretch out across the cell, and you'll have these centrioles on opposite ends. Okay, so that's during prophase. During metaphase, what happens during metaphase is all the chromosomes line up. They line up in the center at a place they call the metaphase plate or the metaphase line, once again depending on the textbook, and you'll see these spindle fibers once again. These fibers here are protein fibers, as I said, and they are a specialized kind of protein called microtubules. There are several different protein structures found throughout a cell. These are particularly the largest ones called microtubules, and they're a protein that is relatively large and very, very long, and they'll stretch from the centrioles of the cell on one end all the way out, and they will anchor themselves to the chromosomes. Typically, they anchor into other specialized proteins found at the centromere. So during metaphase, all the chromosomes line up. Okay, and the meiotic spindle fibers have formed and have connected to, on one side, the centrioles, and on the other side, the chromosomes, okay, at the centromere. And remember that each of these here this is a duplicated chromosome. So it consists of two sister chromatids. So one of these here should be genetically identical to the one on the other side, unless an error occurred during replication. Okay, so other than that, they should be identical to each other. So then what happens is the cell goes through anaphase, and during anaphase, the sister chromatids separate. So as the spindle fibers start to pull back, they actually pull the chromosomes apart, and what was once a duplicated chromosome with sister chromatids, the sister chromatids separate and form individual daughter chromosomes. This is a little bit confusing for people because this over here is a chromosome, but in reality it's a duplicated chromosome with two sister chromatids. When they separate, they become their own chromosomes. So what was once sister chromatids now become daughter chromosomes. So those will separate, and what happens then during anaphase is one set will go one direction and one set will go another direction. During telophase and cytokinesis, what happens, telophase and cytokinesis will sort of lump together, but what you get is you get the nuclear envelope reforming around one set and the nuclear envelope forming around another set, and that cell will then divide into two, and you'll end up with two diploid cells when you're done. Okay, so what you want to do is keep in mind here, you want to make sure you know what happens in each step, but make sure you understand basically what we're doing in this process is we start with one cell. And I don't really like how they've done the chromosomes here, so you can kind of ignore what they're showing you inside the cell. I think could be a little bit confusing. But you start with one cell, you copy everything in it, and then you divide it and you end up with two cells. So the main goal for us, the way we're going to think about this, is that in mitosis what you do is you start with a diploid cell and you end up with two diploid cells. Okay, so if you start with, say, one skin cell, you know, your skin cells or the inside of your cells in your digestive tract, they're always getting damaged and broken, so you need to make new ones. Well, what happens is one of those cells can go through this process of mitosis and can copy everything in one of those cells, and you can make a copy of that cell. And that is essentially what happens in mitosis.